Good morning. Welcome to video number 15. This is gene regulation in prokaryotes. It'll probably be 10, maybe 15 minutes. The eukaryote gene regulation video, the next one, epigenetics really, is going to be the longer of the two. After this video, you should be all right on 2019 number 1. 2019 number 1 was originally a 10-point question. Looks like this. We're mainly concerned this year with A, B, and C. When you get into D and E, it's kind of more like ecology type of stuff. And so that shouldn't be on this year's test. And really what you need this video for is part C. It'll help to have this video under your belt whenever you get to part C. So we're going to look at two main things. We're going to look at positive and negative feedback loops. That's one thing. And then we're going to look at how bacteria regulate their operons, how they decide when to turn genes on and off. So first, let's look at this feedback loop. Let's say that I'm a bacterium and I want to always have a certain amount of tryptophan. So you might remember this example from earlier in the year. Here's my enzyme, and here's an allosteric site. So an inhibitor might bind there. Okay, so this enzyme, its job is to make tryptophan. So 100 enzymes, and they make TRP, tryptophan. Okay, so we're going to be following the right side of the chart first. So if I look at this, it looks like I have a precursor chemical that's shaped like this for this diagram. Enzyme 1 will tweak it, then enzyme 2 will tweak it, then 3, 4, 5, and eventually I end up with tryptophan. All right, makes sense. So I take this, and enzyme 1 apparently does this chemical modification. And then enzyme 2. And then enzyme 3, 4, and 5 very precisely, almost like origami, makes this perfect tryptophan. Okay, great. Let's say I always want about 100 tryptophans. In a human, I always want my body temperature to remain about 98, 99. I don't want my blood sugar to sink down to 10. I don't want it to skyrocket to 500. I want to keep it a pretty steady state. So what if we want to do that with tryptophan? What we'll set up is what's called a negative feedback loop. A negative feedback loop is where whatever is produced by some sort of chain of events, that thing that's produced will turn off its own production. So tryptophan will feed back, making a loop, and it's going to feed back and say stop enzyme 1, which means stop making tryptophan. So again, this process says make tryptophan. What tells it to stop? The presence of tryptophan, right? Kind of makes sense. I might nag my son to clean his room. I go back, it's not clean. I nag him some more. I go back, it's not clean. I nag him some more. What is going to make me stop telling him to clean his room? When I see that his room is clean. So the clean room tells me to quit telling him to clean his room. So a negative feedback loop. Okay, let's say that I have 100 of these enzymes that make tryptophan, and I've made 90 tryptophans. So in theory, 90 of those tryptophans will bind to this enzyme and turn it off. Whenever that inhibitor binds, maybe the enzyme looks like this. So now that enzyme is off. When I take the inhibitor away, now the enzyme is back on. Okay, well in theory, if I've got like 90 tryptophans out there, I know some could still be free floating in space, but in theory, that means that I could have 90 of the enzymes turned off. That means 10 of them are still on, so I'm still making tryptophan. So eventually I get to 100 tryptophans. Now, every enzyme is in theory off. I'm not making any more tryptophans. So when I get to 100, I stop. But now I'm a bacterium, and it's time to build a protein that requires tryptophan. So I'm going to take a tryptophan. So this guy is off because he's inhibited by tryptophan. I'm now going to take that tryptophan away to use it to build a protein over here. When I do, that enzyme is back on. So when I have 99 tryptophans, that means one of the enzymes is on, which means I'll make another tryptophan. So I'll have 100, and then I'll stop. And then I'll use one. So now I have 99, which means I'll make another one. So now I'll have 100. So I always just back and forth, 99, 100, 99, 100. You get too hot, you sweat. You're still too hot, sweat some more. Oh, you're done? Okay, cool. Speaking of cool, now you're too cold, so you shiver. Sweat, shiver, sweat, shiver. Keeps me at a steady state. Okay, now, what if we want to do the same thing from a gene point of view? Now I could maybe like feed back and turn a gene off. That would be accomplishing the same task, but it would be at the level of, instead of protein, at the genetic level. So here is the trip operon. An operon in bacteria is different from eukaryotic genes. Eukaryotic genes are spread out everywhere. They usually have a promoter, one gene, and then a terminator and all the regulatory sequences. With bacteria, they'll tend to have a group of genes with one promoter. So this is trip A, B, C, D, E. That's 1, 2, 3, 4, 5. That codes for these 5 enzymes. So that's gene 1, 2, 3, 4, and 5. Just over here they use letters, A, B, C, D, E. So the idea is, why would I ever, if enzyme 1, 2, 3, 4, and 5 help me to make tryptophan, why would I ever make enzyme 1 and 3 and 5, but not 2 and 4? Right? If I manufacture automobiles, why would I ever make only 3 wheels? That's like, if you're going to make 1, you make 4. And if you make 4 wheels, you make 4 tires, and you make 1 steering wheel, right? Like, you just, if I ever make this part, I would make the rest of it. Okay, so the beginning of the gene is a promoter. A promoter is a parking place for polymerase. So RNA polymerase will land on the promoter. This promoter is currently in the open state. So RNA polymerase can land, read the genes, transcribe them, and we're off. You'll notice over here, though, I've got a repressor. So that repressor doesn't quite fit that operator. The operator is what I'm calling the on-off switch. That repressor doesn't quite fit it, though. It's devil horns. It needs to be field goal. So how can I maybe get that repressor to fit? I got to get it to change shape. That looks like an allosteric site right there. So what if tryptophan fit in there, and now I could turn this set of genes off? That's exactly what's going to happen. When tryptophan hits that repressor, the repressor changes shape, it'll fit into the operator, and now this operon is off. So we now are regulating this gene based off of what we produce. Now let's look at a different situation, and don't worry about calling it positive or negative feedback or anything like that. Let's look at a different situation. Let's say I'm a bacterium, and I can make lactase enzymes. Lactase enzymes help me break down lactose. So if I'm a bacteria and I fall into milk, there's lactose, so I would want lactase enzymes. But if I'm a bacterium and I just fall onto this monitor, there's no milk there. So it's a waste of energy to make lactase enzymes. So what that means is, like, basically I never make lactase enzymes until I fall into some milk or onto some cheese or something else with lactose sugar in it. So my genes are always in the off position until you turn them on. With tryptophan, it was the opposite. With tryptophan, the genes are always on until something turns them off, tryptophan. Here, with the lac operon, it's the opposite. The gene is sort of born, so to speak, in the off position. The repressor fits. So now polymerase can't land on the promoter. What is going to turn this set of genes on? Lactose. So lactose, whenever lactose binds to that repressor, the repressor changes shape and falls out, and now I can start making the lactase enzymes, and those lactase enzymes can start breaking down the lactose. How do I know lactose is there? That's what turned this set of genes on, this operon, what turned them on to begin with. From there, the systems can get more and more complicated. You can kind of have a preference of, like, okay, I'll eat this sugar unless this one's present, then I want to eat this sugar instead. So you can start having more than just a sort of a single file line instead of more like a linear relationship that can get more complicated. But for us, the trip and the lac operon are probably all you need. Thanks for listening. The next video will be eukaryotic gene regulation. After this video, though, don't forget 2019 #1, A, B, and C primarily. Thanks.